Okay, thank you very much for today and good morning. It is my honor to be here and giving this presentation about the project that I've done so far for about 2 years and highly supported by the Asahi Glass Foundation. So the topic that I'm going to talk today is changed a little bit. So I'm going to talk about the osteogenic enhancement of silk fibrin bone scaffolds by forming hybrid composites with bioactive glass through GPTMS during sol gel process. And thank you for the introduction. So I'm Peerapat Thongnuek from Biomedical Engineering Program, Faculty of Engineering. So let's talk about Thai silk. This is the data that I've got from the Queen Sirikit Department of Sericulture in 2008. Sorry for the outdated data, but it is the most recent that I could find. So Thailand exported about 700 tons of silk fabric to worldwide. But we got not only actually, but we got almost 800 million baht back into the country. So this means that 1 kilogram of silk is worth only 1,000 baht. That is approximation. So from my calculation, that is 1 baht, about 1 baht per gram of silk. That is not very good for the effort that our farmers has put into, right? So what if we, sorry, but the screen doesn't fit with my presentation. What if we change our mindset from relying on only the silk fabric? What if we use silk cocoon, silk fibers, and make them into medical device that could increase the value of silk for almost 3,000 bahts per gram of silk, right? So that is a lot of value addition to Thai silk. So as I am in biomedical engineering program, I aim to make medical devices from silk fibrin. So silk fibers, in silk fibers, we have two kinds of proteins. One is silk fibrin, that is account for about 70% of the mass. And another composition is silk sericin, which is about 30% of the weight of silk fiber. And silk sericin is actually the glue that makes silk fibers attached to each other. And the silk fibrin is the structural protein. I'm going to just wait for the adjustment of the screen. So, silk fibrin can be made into bone scaffolds. And what actually are bone scaffolds? So, when we have broken bone, an injury in the bone, and this bone is broken, and the bone is ulnar, for example, so you can see that there is a large gap between these two pieces of the bone. And the problem is that this bone won't heal, or it can heal, but very slowly. And the problem is because the cells in this ulna, in this piece of the bone, cannot communicate well with this piece of the bone. So what we need is the bridge that connects this piece of the bone with this piece of the bone. And the way that the doctor does it is to put bone scaffold into the place. So that allowed the communication between the two pieces of the bone, right? So I did a very similar thing by using silk fibrin to make bone scaffold. And I hope that the Thai silk fibrin can be made into this scaffold and then put into bone fracture and to see how fast the bone can heal itself. But there are some problems that we need to overcome when we use silk fibrin as bone scaffold. So this is the silk fibrin as a bone scaffold, and when we observe the scaffold under scanning electron microscope, we can see that they are very porous, and we have interconnected pores that allow mass transfer, for example, the oxygen and nutrients from one pore into the next pore, and that is very similar to blood transfer in the physiological condition. And The lower picture here is the cross-section of the bone under SEM as well. So you could see that these two structures are very similar, and we hope that the bone scaffold from Thai silk fibrin would behave very similar to the actual bone. But the problem is that when we compare the mechanical property of silk fibrin scaffolds to the cancellous bone, this is the cancellous bone, you could see that the compressive modulus of the cancellous bone is a lot higher. It's like more than 10 times higher than the silk fibrin scaffold. Another problem is that this is the silk fibrin scaffold without any cross-linking. So when I submerge the scaffold in PBS, phosphate buffer saline, for 1 day, what happened here is the degradation. The silk fibrin dissolved in the PBS. A lot of it dissolved, so it loses a lot of mass in 1 day. So it means that the stability of the scaffold is not so good, and the practicality for the doctor is not good either. So we must find something to improve the properties of the silk fibrin. So I looked around and found one material that looks very interesting, and that is bioactive glass. So you could see from the top image here that it's a glass implant. So this piece is a glass implant. It is untreated glass surface, and this is the bone. So you see the gap between this glass implant and the bone, and this gap is actually the fibrous encapsulation. It means that the bone tries to reject the glass implant. But when we change the composition from normal glass to bioactive glass, which incorporates calcium into it, you can see that the gap is very minimal. We cannot see the gap here now. And when we look into it, when we zoom in, 45S5 is the formula name of the bioglass. You could see the formation of this crystal that we call hydroxyapatite, which is a form of calcium phosphate found in the bone. So they think that, this is from this research in 2013, in a review, sorry. So the bioactive glass here that is in the implant structure can allow the formation of calcium phosphate at the interface between the bone and the glass. So this reduces the rejection of the implant. So I think that this bioactive glass thing is very interesting, and what if we incorporate bioactive glass into silk fibrin, that should improve our Thai silk fibrin properties. So the first thing that when I went back to the lab is that I tried to make the composite materials. So the way we did is, I must let you know that to make bone scaffold we need porous structure. And one way of making porous structure is by foaming. So I bought a milk frother. It's the same that we used in the coffee shop. And then we mix the two solutions together and we foam it. You could notice the viscosity of the solution. Initially, it is not very viscous. It's very runny. And then after 20 minutes of frothing, the viscosity increases a lot. And then after that, I put this into the oven, and the foam structure stabilizes itself. And then finally, I got the scaffold. This is the scaffold, the composite scaffold that I got. So this scaffold is made from silk fibrin together with the bioactive glass. So when you look into the microstructure of it, the silk fibrin scaffold has a structure just like this. They are very porous and they have pore interconnection, the same as we found in bioactive glass alone. We could see many pores and interconnected pores. But when we make composite scaffold, you could see the deposition of silk fibrin protein on the wall of the pores of bioactive glass, and that increases the roughness of the pore wall, and that should increase the bioactivity of the scaffold. But there's still a problem. When we mix them together and we got the composite scaffold, and we we did the same. We put the composite scaffold in water, and at the start, the mass is 100%. But one day after that, after submersion, we lost about 65% of the weight. So still, this scaffold wouldn't be very practical in clinic. So we played a little bit with the chemistry of silk fibrin and bioactive glass. So we used a chemical cross-linker that is called GPTMS. It is an organosilane. So it has the a group here, a reactive group here that can react with carboxyl group in silk fibrin. So this reaction would functionalize silk fibrin. And on another end, we have, when there's a presence of acid, we can have hydrolysis of this SiO methyl group. And finally, we got SiOH on this side. And then we can allow them to undergo condensation reaction between calcium silicate network and this functionalized SF, silk fibrin. So finally, we got the hybrid that we have the calcium silicate network forming an intimate connection with the silk fibrin. So this is the hybrid between organic and inorganic network. So we tested for stability again. And what we found is that after one day, silk fibrin lost a lot of the weight. So from 100%, after one day, silk fibrin scaffold lost like 70-something%, whereas bioactive glass is quite stable. And this is when we didn't use the cross-linker. The weight loss is about 65%. And after that, we cross-linked them together, the two networks together. What we got is that after one day, they lost only about 25% of the weight. So the stability is increased by cross-linking with GPTMS. And also, we tested it further by putting them in protease solution. It's a solution that contains an enzyme that can digest protein because silk fibrin is a protein. And we observed that throughout 28 days, so it's almost a month. And what we found is that this is without cross-linking. The lower line is without cross-linking. After 1 month, it lost about 70% of the weight. But after cross-linking, we can maintain 60% of the weight. And that is good because bone non-union could heal for within 3 months. So if we can keep this percentage of the weight after 1 month, we have a high hope that in 3 months, these bone scaffold can maintain its mechanical property and its structure to maintain the bioactive activity of the scaffold. And also we tested for mechanical property of the SF scaffold, SF alone, bioactive glass alone, and the hybrid. So the hybrid exhibited the mechanical property in between bioactive glass and silk fibrin. And this value is actually getting very close to the lower boundary of the cancellous bone. So we quite like that. And we next try to prove that our hybrid scaffold is it has biosafety. So we did cytocompatibility test in accordance with ISO standard. And what we found is that when we extracted the degradation product of the scaffold, of the hybrid scaffold, and we used the extract to culture the cells, the fibroblast cells, and what we found is that the survival of the cell in the media containing the degradation product of the hybrid is very close to the control. This means that our hybrid is safe. And then we try to prove that our hybrid scaffolds can exhibit bioactivity or bone forming potential. So we put them in the simulated body fluid. It's the fluid that has the same concentration of electrolytes in the physiological condition. So what we found is that after 9 days of submersion, we found this cloudy layer. So we thought that there might be some crystallization of ions. And what we found is that when we collected the crystal, and also the coagulation on the surface of the scaffold, and we proved that there is a concentration of calcium and phosphate in a very similar ratio to the hydroxyapatite. So we concluded that our hybrid scaffold can form hydroxyapatite, the same as in the bone. And also, we tested for the early marker for osteogenesis and late marker. And we found that our hybrid scaffold has the better osteogenic property compared to the silk fibrin. So this means that the hybrid scaffold showed in vitro osteogenicity greater than that of silk fibrin scaffold. And before I end my presentation, I would like to thank Asahi Glass Foundation because of it. So there are 3 outputs that I've got from this project. The first one is the prototype that I can do more study on it. This is the hybrid scaffold made from silk fibrin and bioactive glass, and also a publication in a journal, and also people. So this fund gives me opportunity to push these 2 students to get their degree in biomedical engineering, including Pimnara, Wattanachai, and Juthatip. So finally, I would like to thank the Asahi Glass Foundation, Biomedical Engineering Program, and Biomaterial Engineering for Medical and Health Research Unit and Chulalongkorn University. Thank you.
Thank you very much for a nice talk.